Good afternoon, everyone. Welcome you all for the second session on water quality. That is exclusively, we will be knowing about the biological water quality and then followed by the water treatment. So biological parameters, as we know that in water there will be thousands of biological species, okay, which may enter through the various sources. So which mainly consists of, we can say, phytoplankton, diatom, and dinoflagellate, zooplankton. It can be protozoa, bacteria, fungi, and viruses. Okay, so these biological organisms, based on how what are the effect it will going to cause, so we call most of them as the pathogens. Okay, so pathogens are the microorganism that cause the diseases. So there are various types of the pathogens which are very concerned for us. One is bacteria, then viruses, protozoa, helminthus. Okay, so this is a pic which shows the whatever the number of the bacteria that we may find on the tip of a pin. Okay, so there are so many bacteria will be there in a small point. So these microorganisms will going to cause various health effects. Okay, so when we consider the size of these microorganisms, so the smallest microorganism is virus, and then comes the bacteria, and the bigger one is the protozoa, and the largest is the helminthus. Okay, this is the order that we will going to find. So the virus will be usually in the size range from zero point 0.02 to zero point 0.2 microns, and the bacteria is zero point 0.2 to 5 microns, and the protozoas will be from 4 to 20 microns, and the helminthus will be about 40 to 100 microns. Okay, so usually in the sand filters, either it can be rapid sand or the slow sand, which we will be using in the water treatment. These sand filters pore size will be about 1 micron. Okay, that we can say some bacteria which are having size greater than the 1 microns, so we can able to filter, or we can able to remove during the filtration process. Okay, so we can overcome the problem of the helminthus, the protozoa, and the bacteria which are having the size more than 1 microns can be removed. Okay, so viruses which causes the infections are important, is the hepatitis, dengue fever, and the polio. And as you all aware that the virus will going to depend on the host cells if they want to grow, that they infect the they will going to infect the host cell when they are reproducing or when they replicate. So when simulated, new viruses will going to form, and they will going to burst out of the host cell, and they will going to kill that host cell, and then they will going to infect the other cells. Okay, so some viruses can remain viable outside of the host cell. Okay, very few, not all the species, for a longer period. And also some viruses are even have a capacity to sustain themselves even in the dry condition. Coming to the bacteria, cholera, salmonella, shigella, typhoid, trachoma, these are some important diseases which are caused by the bacteria. This is the most dominant microorganism which is found in the feces of the animals, that is especially the warm-blooded animals. So most diverse, as I told, it is the most diverse group of microorganism, and we can say it is the most simplest living organism which is there in our life system. So in a feces, we can say just in 1 gram of the feces, we can have the billions of the bacteria. So factors that affect the number and the type of the bacteria in the water, so mainly depends on these factors. One is the type of the water, whether it is surface or it is from any springs. Then the presence of the organic matter, temperature, light, pH, dissolved oxygen, and the type of the filtration that we have used. So next organism is the protozoa. So these protozoas are single-celled organisms. It is able to form the cysts, which can be resistant to the chlorine most of the time, and some even can stay alive outside of the host cell. Okay, in case of the viruses, we it needs a host cell for its survival most of the time. Very rare viruses can able to sustain outside. So here protozoas also can sustain alive outside the host cell. Okay, the most important protozoas are Giardia and Cryptosporidia. So next is Helminthus. Okay, under Helminthus we have the various worms: round worm, hookworm, guinea worm, then bilharzia. Okay, these are some important Helminthus which are usually found in the warm-blooded animals' intestines. So they can live for many years in the body, and most of these helminths will not going to multiply within the host cell. Only it will going to derive its energy from the host. Okay, so most helminths are going to pass out of the body through the feces. So, as we have observed that water source is mainly polluted because of this feces, warm-blooded animals' feces. So, this feces will going to introduce a variety of the pathogens. Okay, these are the bacterial infections which are caused or which may be spread through the feces. Then the viral hepatitis A, E, rotavirus, and then the poliomyelitis, then the helminthus. So these are the helminthus which will going to cause the various disease in human, and the protozoa, it is amoeboisis, giardiasis, and uh, balantiardiasis are the protozoal infections. So, to know whether the water contains these microorganisms or not, we need to carry out the microbiological tests. So, testing for every pathogen, that is depending on what type of microorganisms that we need to test. So, based on that, the time that is taken will going to vary, and most of these testings are expensive in nature. So, in generally for the water, we will going to do the bacterial test most commonly, which we call as the bacteria as the indicator organism. The tests for the bacterial indicator organisms, as we do very frequently, are most commonly used because it is cheaper in nature, easier to perform, faster results when compared with the other microorganism, and does not require highly trained personnels. So with simple knowledge, a person can lab personnel can able to do this analysis. Bacteria, when we say the bacteria, we have the various types. So for our importance, E. coli, okay, that is presence of the E. coli will going to indicate the fecal contamination, whereas thermotolerant coliforms, which are usually found in the intestines of warm-blooded animals, and then we conduct the total coliform tests. Okay, the presence of this total coliform in water will going to indicate the contamination of that water, and we may have these coliforms which can be either heterotropic in nature. Okay, so we generally call the bacteria as the indicator organism. Means an indicator organism is one whose presence presumes that the contamination has occurred and suggests that the nature and the extent of the contamination of the water has to be checked. Okay, so most of the time we will going to check for E. coli, that is Escherichia coli, then the fecal coliform, fecal streptococci, and then the Clostridium perfringens. Okay, so the presence of the fecal streptococci along with the coliforms in the absence of E. coli is also confirmatory of the fecal pollution. So the two methods, testing methods that are commonly adopted in water tests are: one is membrane filtration technique, the other one is multiple tube fermentation technique. Membrane filtration technique. So here a membrane, that is a thin material, is used, okay, that has the pores of a specific size. So the membranes will going to trap the larger particles that won't allow through the pores of those membrane, and letting the water and other smaller substances to pass through. Okay, so this is the schematic sketch how the membrane filtration will be. In this membrane filtration, it is the most accurate method to count the bacteria. And here we will filter 100 ml of the water sample using a filter paper, and we add the broth to a petri dish which provides the nutrient for the growth of the microorganisms which we want to detect. Then filter the water using the filtration equipment, and then transfer the filter paper to the petri dish, and then we will going to incubate for 24 to 48 hours depending on the whatever the nature of the broth that we will be using. If the indicator organism is present, then the colonies will going to appear over the filter paper and can be counted. The results are reported most as the number of colonies per 100 ml. Okay, that is CFU per 100 ml. Colony forming units per 100 ml. So that is how we will going to report the results of this membrane filtration. Okay, this is the sketch showing the various apparatus that we will be using. So this is the filter unit, okay, membrane filter unit, which we will be employing to filter the water sample. So we will going to place the filter paper here, okay, over this MFT apparatus, and then in this funnel we will going to pour the 100 ml of the sample, and this apparatus will be connected to a vacuum pump. Okay, because the smaller the particle size, that will going to clog the pores of the filter paper, so water will not going to pass through easily. So with by creating the section here, we can able to make the water to pass through that filter paper, and we can retain the particles or the microorganisms which are greater than the whatever the pore size of the filter paper that we have taken. Okay, and then after filtration of 100 ml of that sample, that filter paper is transferred to the petri dish, which for which we already add the broth. Okay, depending on what type or what species of the microorganisms we want to detect, so we will be taking that particular broth, and over that broth we will going to place this filter paper, and then we will going to incubate at 35 degrees centigrade. For initially we will going to check after 24 hours. If we won't find any colonies over that filter paper, then we can incubate for further 24 hours, that is up to 48 hours we can wait. And then if we found the dots like this, so which we call as the colonies, so we can able to count that and express as, so each dot we will be considering as the colony forming unit. So we will going to express whatever the number we got as CFU per 100 ml. The next method is the multiple tube fermentation technique. So in this, coliforms are known to ferment the lactose. Okay, that is the one of the important feature of the coliforms. So here, keeping that feature in the mind, we will be employing the various types of the lactose broths in this test. So initially, the lactose broth is placed in a series of test tubes and inoculated with a decimal fraction of sample. So here we will be taking the various decimal fractions. We will be taking 10 ml, 1 ml of sample, zero point 0.1 ml of sample, and 0.01 ml of the sample. So in a number of series. So first stage of the procedure is referred as the presumptive test. Okay, here in the presumptive test, the broth that we will be using is called as L'Oreal tryptose broth. Okay, we will be using the L'Oreal tryptose broth. The tubes with the gas developments are presumed to have the coliforms. Okay, so we will add the sample to this L'Oreal uh, tryptose broth tubes, and then we will plug it, and we will going to allow for about 24 hours at a temperature of about 35 degrees centigrade. After 24 hours, if we have if we found that the gas is formed or gas is produced in that particular tube, then that is going to confirm us the coliform is present in that sample. So we will going to next continue with the same test. Okay. So second phase is called as the confirmed test. Okay. Second phase of that MTFT is referred as the confirmed test, where we will be using the brilliant green lactose bile broth. Okay, so this confirm test is again we conduct to know or to actually confirm ourselves whatever the gas release in the test tubes is mainly due to the microbes or not. Okay, so that's why it is referred as the confirm test. So in this confirm test, we will be taking the whatever the samples which are which are considered to be positive. Okay, so those test tubes will only consider for the next test. And after once we got the confirmation, that is again if the gas is formed in the test tubes, so then we move on to the third phase. So that third phase, or which is also called as the last stage of the test, is known as the completed test. Okay, so in this completed test, instead of using the broth, we will be using the agar, so which we call as endo agar, or it is another name we call that as EMB agar. So based on this tests, okay, we will be calculating, that is MTFT, we will be calculating the most probable number, or MPN. So this most probable number will going to provide the information on the number of bacteria that are most likely to be present in that water sample. So we need to add the water sample or the diluted sample to at least 5 test tubes. Okay, so and also the various dilutions we need to consider. And once we take the various dilutions, 5 test tubes, we need to incubate for 24 hours. Okay, if we won't get the results in 24 hours, we need to continue for 48 hours. Then the gas production or the cloudiness in that sample, uh, then that will going to indicate the presence of the bacteria. And using a table which is going to provide it, we will going to report what is the probable number of organisms based on the number of tubes which are got to be positive. Okay, so this is again a schematic outline how this multiple tubes test takes place. So this is the presumptive test. Okay, first stage, we use the lactose bile broth. Then we incubate. We after adding the sample at the various dilutions, we incubate for about 24 hours at 35 degree. If gas is produced, then we consider that as the positive test. If no gas is produced, then we will continue for next 24 hours, and then we will check whether the gas formation is observed or not. Okay, again after if we found that gas is formed, then we will going to consider that as the positive test. So, okay, this is how the presumptive test tubes will be. So this like bubble formation within the Durham's tube of the test tube. So if we got the bubble like this, then we will going to consider that as the positive ones. So this is the schematic showing the serial dilutions that we will be taking in this test. Second phase is the confirm test. So in the confirm test, we will be instead of taking the L'Oreal tryptose broth, we will going to take brilliant green lactose bile broth, and then we will add the samples. Okay, then if the gas is produced after 24 hours, then that is considered as positive. If no gas is formed, then it will be continued for further 24 hours, and then it will be checked for the presence or the formation of the gas. Okay, so if the no gas is produced, then it is considered as the negative test. If gas is produced, then that will be considered as the positive test. Then it will be taken to the next stage of test. This is how the brilliant green lactose bile broth will be. So this is the tube. Okay, in these three test tubes, the two test tubes are considered as negative because we can't see any gas formation here in the Durham's wheel. Whereas in the third one, we can see the gas bubble or gas is collected in this Durham's wheel. So that we can consider as positive. So last stage is the completed test. So here, whatever the test tubes which are considered to be positive are taken to the next phase. So where the sample from the test tube will be added over the endo or the EMB agar, and then it will be incubated for 24 hours at 35 degrees centigrade. Okay, based on that number of colonies that will going to form, we will going to count that, and then we will going to express that as CFU per 100 ml. Okay, this is how the colonies that will going to form over the EMP agar. Okay, so while adding the sample over this EMB agar, we can follow either the pour plate method or we can follow the streak plate method. So we can calculate using the Thomas equation, we can calculate the most probable number. If we won't have that table, then using this Thomas equation, we can calculate using the number of positive tubes and the number of negative tubes. The equation is MPN value will be equal to number of positive tubes into 100 divided by square root of ml of sample in negative tubes into ml of sample in all tubes. Okay, so this will going to give us the quantitative results, and it is simple to understand and use, and it is relatively inexpensive, so can be used with the turbid water. But while taking the dilutions and assessing the formations of the gas needs some training, and also it requires the incubator and other accessories. Okay, so now so this is again one of the important questions you may get, because with respect to the public health, it is very much important as an engineer to check the most probable number of the organism that may be present in that sample. So they will give this question: a water sample is tested for the presence of the coliform organism by MPN method. So this is the whatever the test results they have got, and we need to use the Thomas equation to calculate the MPN value. So size of the sample is, these are the different sizes. They have considered the sample, and these are the number of positive tubes and number of negative tubes. Okay. So when we take consider this, number of positive is 4, number of negative is 1. So totally they have considered the 5 test tubes for each sample size. Okay. Now, let us substitute the value in the Thomas equation. So, Thomas equation is number of positive tubes. So, we need to take the total number of the positive tubes. So, 4 plus 4 plus 2 is 10. So, 10 into 100 divided by square root of ml of sample in the negative tubes. Negative tube is 1 into 10, so that is 10. 1 into 1, that is 11, and 1.1 into 3 plus 0.01 into 5. Okay, so when we add all these, so we get the ml of sample in the negative tubes as 11.15 into ml of sample in all the tubes. Okay, total number of tubes that were taken for the each sample size is 5. So what we will going to do is 10 into 5 plus 1 into 5 plus zero point 0.1 into 5 plus 0.01 into 5. So totally we get 55.55. Okay, that is the ml of sample that we have taken in all the test tubes. Okay, when we simplify this, we will going to get the value as 40.18. Okay, so that we can express as, so we will going to neglect the decimal value, so we consider the whole value. So it is 40 number of organisms are present per 100 ml of that sample. Okay, so this is the result we got for this particular test. Okay, like that we can easily calculate what were the most probable number of organism that is present in the given water sample. Okay, so similarly, a water sample is analyzed for presence of coliform using MTFT. Okay, these are the results. Positive is 5, negative 0. Okay, here the dilutions is different. Okay, depending on this dilutions or whatever the samples sizes that we are taking, the value will vary. So MPN of coliform have to be obtained for the above results using the MPN index. Okay, so MPN of the coliform per 100 ml is how much? Okay, they are asking. So for this MPN index, so various combinations of the positive results need to be considered. So when 5 test tubes used per dilution of 10 ml, 0.1 ml and 0.1 ml. So these are all the combinations they have given. 0 to 4, then MPN index. Okay, this is a ready reckoner table. For this 0 to 4 positive results, the index is 11, 135, 19, 420, 22, 531, it is 110. So we here we got the positive result as 531. So based on this, the combination of the 531, that is, this is our combination. So MPN index is 110 per 100 ml. So we will be taking the option A, sorry, option answer is the option D. So how we calculate? So we have taken the sample sizes 0.01, 0001, 0.00001, which is 1000 times lesser than the standard sample sizes. Okay, this is the standard sample size 10, 1 and zero point 0.1. But here we have got for the whatever the results that we got is for 0.01, 0.001 and 0.0001. So based on that, we got the from this test MPN index, we got the value as 110. So 110 into 1000 we need to multiply. So we will got the value as 1,10,000. Okay, or 110,000 is the whatever the value that we will be getting for this combination. So next, we will talk about the guidelines and the standards. So because before we move on to the water treatment, so let me talk about what is the difference between the guideline and the standard. So guideline is a recommended limit that should not be exceeded, whereas the standard is a mandatory limit that must not be exceeded. Okay, so usually WHO guidelines are used for the drinking water, whereas the guideline value is just to ensure the safety of the drinking water, whereas standards may vary among one country to another region. So in India, we follow the Bureau of Indian Standards, so that is IS 10,500, and the latest given standard is in the year 2012. Okay, we will be following this IS 10,500 2012. Okay, these are all the drinking water standards. So next, coming to the doing this all these tests, the purpose of examination of the water is to detect and assess the degree of the fecal pollution, and then to assess the amount of treatment that we need to render to that water source so that we can make that water safe for consumption, and also to ascertain the efficiency of the purification treatment at the various stages, and then to locate and identify the cause of deterioration of water quality, and then to establish the purity of the final water as it leaves the purification work. Okay, next, to proceed with the water quality index, so we will know about the water quality index, that is WQI. A water quality index is a means by which the water quality data is summarized for reporting to the public in a consistent manner. The water quality index may be defined as the rating that reflects the composite influence of a number of water quality factors on the overall quality of water. So here, water quality index is also defined as it's a means to summarize large amounts of water quality data into simple terms for reporting to management or to the public to make a decision. So it will be easy for the authorities to make the decision based on this WQI. The advantages of an index includes its ability to represent the measurements of a variety of the variable in a single number, and this WQI is one of the effective tool to communicate the information about the quality of the water either to the concerned authorities or to the citizens. And this water quality trend, uh, to know the water quality trend also we can able to use this WQI, and then to implement the water quality monitoring program or the water quality improvement program very effectively. So we can use the WQI as the basis. So the various methods are used to assess the WQI. So these are the various WQIs that are being used. So one is National Sanitation Foundation Water Quality Index. Okay, so a usual water quality index method which is developed by National Sanitation Foundation. So in this method, 9 water quality parameters like temperature, pH, turbidity, fecal coliform, DO, BOD, and phosphate, nitrate, and solids is considered. Okay, please remember, they may ask the question in case of NSF WQI, what is the number of water quality parameters they have considered, or they may give the parameters and different combinations, and they may ask you to select the correct 9 water quality parameters which are considered. Okay, so here water quality data are recorded and transferred to a weighting curve chart where a numerical value of WQI is obtained. Okay, this is the formula they will be using to get that numerical value. So based on that numerical value, the index limit, they will be giving the whatever the ratings. So 90 to 100, it is excellent quality. 70 to 90, good. 50 to 70, moderate. 25 to 50, it is bad. And 0 to 25, it is considered as very bad quality. Okay. Next is CCME, that is Canadian Council of Ministers of the Environment Water Quality Index. So this particular model will going to provide a very consistent method. So which is formulated by the Canadian Council of Ministers of the Environment. Okay, we call that as CCME. And here the index is based on the combination of the three factors. Okay, the three factors, we say F1, F2, and F3, where F1 is the number of variables whose objectives are not met, and the F2 is the frequency with which the objectives are not met. Okay, that is considered as F2. And the third one is the amount by which objectives are not met. Okay, that is F3. Okay, these values are calculated using these three relations. F1 is number of failed variables by total number of variables into 100. F2 is number of failed tests by the total number of tests into 100. So here the test value which are not exceeding the test objective. Okay, usually we will going to consider the standard value as our objective values. So when we carry out the test for a particular parameter, whatever the test values that are not exceeding, okay, that we need to consider here. So excursion value is calculated. So failed test value divided by the objective minus 1. Okay, that will gives us the excursion. The test value not falling below the objective is calculated using this relation. Objective divided by the failed test value minus 1. So that we will be calculating, and using these excursion we will be calculating the NSC, that we will going to divide it by whatever the number of tests that we conduct. Okay, then using this NSC value we will be calculating the F3 NSC by 0.01 NSC plus zero point zero 0.01 into 100. We will going to get the F3 value, and then we can calculate the CCME WQI using this relation: 100 minus F1 square plus F2 square F3 square whole to the power of half, or we can say square root of F1 square F2 square F3 square divided by 1.732. And this is the rating they will going to they have provided, CCME has provided for the various WKI values. Okay, 95 to 100 is excellent, 80 to 94 is good, fair is 65 to 79, and 44 to 64 is the marginal one, and 0 to 44 is poor. Okay, so similarly Oregon has given one water quality index. So this Oregon water quality index creates a score to evaluate the general water quality. Especially it has been devised for the exclusively for this Oregon stream. So the parameter that covered in this method are these: temperature, DO, BOD, pH, ammonia, nitrate, nitrogen, total phosphorus, total solids, and the fecal coliform. Okay, this model is designed based on the whatever the National Sanitation Foundation has derived, where the along with that the Delphi method was used for the variable selection. Okay, so that is the important feature of this Oregon WQI. So index employs the concept of harmonic averaging. So here, if it is less than 60, then it is considered as poor. 60 to 79 is less than 60 is very poor, 60 to 79 is poor, and 80 to 84 is considered as fair, and if it is 85 to 89, then that quality is considered as good. More than 90 to 100, it is excellent. Okay, the last method, that is weighted arithmetic water quality index method. So in this method, this is the widely used method by the various scientists or the researchers. So here in this method, it is classified the water quality according to the degree of the purity by using most commonly measured water quality variables. So this is the formula they have used in calculating the WQI: sigma Wn into Qn divided by sigma Wn, where Wn is K by Sn. Sn is the standard permissible value of the nth parameter, and Qn is calculated using the relation 100 into Vn minus VIO divided by Sn minus VIO, where Vn is the estimated value of the nth parameter of the given sampling station, whereas VIO is the ideal value of the nth parameter in pure water. And K value is calculated using the relation 1 by 1 divided by 1 by sigma Si, where the I value will vary from 1 to n, and Si is the standard permissible value for the nth parameter. Okay, so using that we will be calculating the weighted average WQI. So here, if the it is the rating will going to give the reverse status. So 0 to 25, it is considered as the excellent water quality. So greater the WKI value, the worsen is the quality of that water. Okay, so 26 to 50, good water quality; 51 to 75, poor; 76 to 100, very poor; more than 100, it is unsuitable for drinking. So now to talk about the water treatment. So before we go in depth, let me say the objectives of water treatment: to remove the color, dissolved gas, and cloudiness; to remove the objectionable taste and odor; to remove the disease-producing microorganisms; to remove hardness; to make the water suitable for various variety of purposes, maybe for steam generation or other industrial activity, dyeing, brewing, so on. Okay. So this is a conventional flow chart, which is generally used in a water treatment plant. So raw water, which is next taken to from the source, will be pumped to the aeration tank. Then addition of the alum, that we call as the coagulation. Then the flocculation tanks. From the flocculation tank, we will be taken that water to the sedimentation tank. Then that sedimented treated water will be taken to the filters, sand filters. That is, rapid sand filters is the most commonly used one. Then from the filtration, the water will be next taken to the chlorination, and after the chlorination, the water will be stored in the water reservoir, and then it will be supplied to the public. Okay, so this is how the various treatment units are used in a conventional water treatment plant. So again, this is the flow sheet. So here, from the screening, aeration, depending on the water quality, they can go for sedimentation with coagulation or without the coagulation, filtration, disinfection, followed by the miscellaneous method. So treatment methods are classified as unit operations and unit processes. Unit operations are those treatment methods where the physical forces will going to predominate, whereas the treatment methods where chemical are used or the biological organisms are used, so we call those treatment methods as the unit processes. Okay, so water treatment methods that is commonly used are screening, aeration, excuse me, excuse. So screening, aeration, sedimentation with or without coagulation, filtration and disinfection. So one simple example question, which is from the previous year question paper. The potable water is prepared from the turbid surface water by adopting the following treatment sequence. Okay, these are all the options they have given. So we need to select the one which is correct. Okay, so in this first option, turbid surface water, coagulation, flocculation, sedimentation, filtration, disinfection, storage. Whereas in the second option, they have given turbid surface water, disinfection, which is wrong. Turbid surface water, filtration is also wrong. Turbid surface water, sedimentation is also wrong. Okay, these four options, the correct sequence of the water treatment is the first one, that is turbid surface water, coagulation, flocculation, sedimentation, filtration, disinfection, and storage and supply. Okay, the answer is A. So now, before I will stop here so that we can uh, start in the next session completely with the treatment methods. Okay, we will study in detail about the treatment methods.